Today, we are diving deep into a hidden world, a molecular battlefield that's playing out inside millions of people every single day. We're talking about herb-drug interactions, where ancient remedies and modern pharmaceuticals collide. This isn't just theory, it's a reality with huge implications for global health. So just how big is this? Well, the World Health Organization figures that over 80% of the world's population uses some form of complementary and alternative medicine. This isn't a niche practice. It creates this massive, often invisible overlap with prescription drugs, especially in high-stakes areas like cancer treatment, where more than half of patients might be combining herbs and drugs, often without their doctors even knowing. And that lack of communication, it leads to a shocking documentation gap. Get this. A hospital audit in Liverpool found that while 83% of patients using herbal remedies were also on prescription drugs, their herb use was documented in their clinical records only 1% of the time. Just 1%. We are basically flying blind in this massive data void. So that brings us to the big question, right? What is actually happening inside this unmapped molecular battlefield? You have hundreds of active compounds from a single herb meeting a single powerful drug molecule. The result? A network of interactions that can be wildly unpredictable. Sometimes it's helpful, but other times it can lead to total therapeutic failure or worse. Okay, so here's our game plan. We'll start with the scale of this hidden epidemic. Then we'll step onto what's called the ADME battlefield to see the core mechanics. After that, we'll look at the even bigger picture of complex network warfare, check out the real-world outcomes, see what tools we have to fight this, and finally, peek into the future of personalized prediction. All right, first things first. To really get a handle on this issue, you have to understand the fundamental difference between these two types of medicines. It's where the whole problem starts. And here is the absolute core of the complexity. Think of a conventional drug as a sniper. One molecule, one specific target. In herbal medicine, on the other hand, is like a whole army, a complex cocktail of phytochemicals. Because you have so many active components, the chance of a significant herb-drug interaction is just way, way higher than a drug-drug interaction. The numbers really drive this home. 77% of people using alternative medicine are using herbs specifically. And what's really concerning is that research shows almost half of them, 46%, are taking something with known side effects or interaction risks. And this risk just gets magnified by the fact that nobody's writing it down. So how do these interactions actually work? To understand that, we need to enter what scientists call the ADME battlefield. It's basically the journey any foreign substance or a xenobiotic takes through the body. Now, an interaction can happen at any of these four stages: absorption, distribution, metabolism, or excretion. But the main front line, where most of the action happens, is metabolism. Any interference here, mostly in the liver and the gut wall, can totally change how much of a drug gets into your system. And it's all thanks to a very important family of enzymes. The MVPs of this metabolic battlefield are the cytochrome P450 enzymes, or CYPs. And what's incredible is that just 3 of these subfamilies, CYP3A4, 2D6, and 2C9, are doing almost all the heavy lifting. They're responsible for breaking down nearly 95% of all drugs we use. So you can see how just one compound from an herb, messing with one of these enzymes, can cause a system-wide ripple effect. Herbs can mess with these enzymes in 2 main ways: induction or inhibition. With induction, like we see with St. John's wort, the enzyme gets sped up. It metabolizes the drug too fast, and the drug might not work at all. Inhibition is the opposite. It slows the enzyme down, leading to a buildup of the drug, which can become toxic. Either way, you've knocked the drug out of its safe and effective zone. And the clinical evidence for this is, well, it's pretty sobering. St. John's wort can slash levels of the crucial anti-rejection drug cyclosporin by up to 62%, putting a transplant patient at risk. A compound in garlic can directly inhibit the CY2E1 enzyme. And we've all heard about grapefruit juice. It can block transporters and cause dangerous spikes in drugs like statins. But these one-on-one clashes are just part of the story. Now we have to zoom out from these single enzyme effects to what researchers are calling complex network warfare. This is where the entire physiological environment of the patient gets involved. This is modeled by something called the CHNET interplay. Yeah, it's a mouthful, but it stands for cytokines, hormones, neurotransmitters, enzymes, and transporters. This web of molecules keeps our bodies in balance. But here's the key part. A disease state, like diabetes or cancer, messes up this network, and that completely changes how a patient responds to everything, even before an herb and a drug interact. This means that the battlefield is totally unique in every single patient. An underlying disease can change which enzymes are active. Your personal genetics create huge differences in how fast you metabolize things. Add in other medications, or even just systemic inflammation, and the whole landscape shifts. This is why you can't just rely on data from healthy volunteers. So, what does this all mean for patients in the real world? This is critical. The outcomes aren't just bad. They span a full spectrum, from harmful to helpful. And the whole goal of integrative medicine is to figure out how to predict and avoid the harm while actually using the helpful interactions. This table, which is based on dozens of real clinical cases, shows that whole spectrum perfectly. An interaction can be fatal, like St. John's wort and SSRIs causing serotonin syndrome, but it can also be incredibly therapeutic. You have herbs like Withonia somnifera that can actually make chemotherapy work better, or Tenespora protifolia, which can reverse some of chemo's nasty side effects. Warfarin is a perfect example of this variability. It's a blood thinner with a really narrow safety window, and its effect, which we measure with something called INR, can be thrown way off by common things. Look at this, a 30% change with cranberry versus an almost 70% change with soy milk. Both of those can create huge clinical risks, from blood clots to severe bleeding. Okay, so the problem is enormous and incredibly complex. So how on earth can we possibly start to predict and manage all of this? Let's pivot now from the problem to the solutions and take a look at the modern research arsenal being used to map this battlefield. The modern strategy follows a really smart, logical hierarchy. It all starts in silico. That means on a computer, using modeling and virtual screening to predict which interactions might happen. Then, the promising candidates move to in vitro testing in the lab, using things like human enzyme kits. And finally, the most important step is in vivo validation in actual clinical trials, often using really clever cocktail probe strategies in humans. And none of this research would be possible without a whole bunch of specialized databases. You have things like SuperSciAp, which tracks thousands of specific enzyme interactions. India's traditional knowledge digital library, the TKDL, is this amazing resource that catalogs traditional formulas. And then you have comprehensive platforms like DIDB that are trying to pull all the data, drug, herb, food, and disease, into one place to build a truly holistic map. And that brings us to the final frontier. Where is all of this incredible research headed? Well, the ultimate goal is to move beyond those generic warning labels you see on bottles and towards a future of truly personalized, predictive models for every single patient. And there are some really cool new ideas emerging to get us there. There's the whole system strategy, which looks at the entire herbal extract instead of trying to isolate single compounds. Researchers are building complex drug, disease, herb network models. They're integrating data from our microbiome, those 100 trillion gut microbes that also affect drug metabolism. And they're even developing frameworks like Ayurvedization to bridge the gap between traditional wisdom and modern science. And really, that's what it all comes down to. The future of integrative medicine depends on us achieving a computational mastery of these incredibly complex molecular networks. This molecular battlefield is, without a doubt, a huge clinical risk. But it's also our greatest therapeutic opportunity. The chance to finally harness all this complexity to deliver truly personalized and effective healthcare.